Development of new drugs. Since the discovery of antibiotics, research and development (R&D) efforts have provided new drugs in time to treat bacteria that became resistant to older antibiotics, but in the 2000s there has been concern that development has slowed enough that seriously ill people may run out of treatment options. 238-239 Another concern is that practitioners may become reluctant to perform routine surgeries because of the increased risk of harmful infection. 240 Backup treatments can have serious side effects. For example, antibiotics like aminoglycosides, such as amikacin, gentamicin, kanamycin, streptomycin, etc. used for the treatment of drug-resistant tuberculosis and cystic fibrosis can cause respiratory disorders, deafness and kidney failure. 241-242 The potential crisis at hand is the result of a marked decrease in industry research and development. 243-244 Poor financial investment in antibiotic research has exacerbated the situation. 245-243 The pharmaceutical industry has little incentive to invest in antibiotics because of the high risk and because the potential financial returns are less likely to cover the cost of development than for other pharmaceuticals. 246 In 2011, Pfizer, one of the last major pharmaceutical companies developing new antibiotics, shut down its primary research effort, citing poor shareholder returns relative to drugs for chronic illnesses. 247 However, small and medium-sized pharmaceutical companies are still active in antibiotic drug research. In particular, apart from classical synthetic chemistry methodologies, researchers have developed a combinatorial synthetic biology platform on single-cell level in a high-throughput screening manner to diversify novel anthopeptides. 248 In the 5 to 10 years since 2010, there has been a significant change in the ways new antimicrobial agents are discovered and developed, principally via the formation of public-private funding initiatives. These include CARB-X, 249, which focuses on non-clinical and early-phase development of novel antibiotics, vaccines, rapid diagnostics, novel gram-negative antibiotic, GNA Now, 250, which is part of the EU's Innovative Medicines Initiative, and replenishing and enabling the pipeline for Anti-Infective Resistance Impact Fund, REPAIR. 251 Later-stage clinical development is supported by the AMR Action Fund, which in turn is supported by multiple investors with the aim of developing 2 to 4 new antimicrobial agents by 2030. The delivery of these trials is facilitated by national and international networks supported by the Clinical Research Network of the National Institute for Health and Care Research (NIHR), European Clinical Research Alliance in Infectious Diseases (ECRAID), and the recently formed ADVANCE-ID, which is a clinical research network based in Asia. 252 The Global Antimicrobial Research and Development Partnership (GARDP) is generating new evidence for global AMR threats such as neonatal sepsis, treatment of serious bacterial infections and sexually transmitted infections, as well as addressing global access to new and strategically important antibacterial drugs. 253 The discovery and development of new antimicrobial agents has been facilitated by regulatory advances, which have been principally led by the European Medicines Agency (EMA) and the Food and Drug Administration (FDA). These processes are increasingly aligned, although important differences remain and drug developers must prepare separate documents. New development pathways have been developed to help with the approval of new antimicrobial agents that address unmet needs, such as the limited population pathway for antibacterial and antifungal drugs (LPAD). These new pathways are required because of difficulties in conducting large definitive phase 3 clinical trials in a timely way. Some of the economic impediments to the development of new antimicrobial agents have been addressed by innovative reimbursement schemes that delink payment of antimicrobials from volume-based sales. In the UK, a market entry reward scheme has been pioneered by the National Institute for Clinical Excellence (NICE), whereby an annual subscription fee is paid for use of strategically valuable antimicrobial agents. Spiderocol and ceftazidime aviabactam are the first agents to be used in this manner, and the scheme is potential blueprint for comparable programs in other countries. The available classes of antifungal drugs are still limited, but as of 2021 novel classes of antifungals are being developed and are undergoing various stages of clinical trials to assess performance. 254 Scientists have started using advanced computational approaches with supercomputers for the development of new antibiotic derivatives to deal with antimicrobial resistance. 244, 255, 256. Biomaterials. Using antibiotic-free alternatives in bone infection treatment may help decrease the use of antibiotics and thus antimicrobial resistance. 36 The bone regeneration material bioactive glass S53P4 has shown to effectively inhibit the bacterial growth of up to 50 clinically relevant bacteria including MRSA and MRSA. 257, 258, 259. Nanomaterials. During the last decades, copper and silver nanomaterials have demonstrated appealing features for the development of a new family of antimicrobial agents. 260 Nanoparticles, 1-100 nanometers, show unique properties and promise as antimicrobial agents against resistant bacteria. Silver, NPs, and gold nanoparticles, are extensively studied, disrupting bacterial cell membranes and interfering with protein synthesis. Zinc oxide, ZNO-NPs, copper, CuNPs, and silica, SynPs, nanoparticles also exhibit antimicrobial properties. However, high synthesis costs, potential toxicity, and instability pose challenges. To overcome these, biological synthesis methods and combination therapies with other antimicrobials are explored. Enhanced biocompatibility and targeting are also under investigation to improve efficacy. 232. Rediscovery of ancient treatments. Similar to the situation in malaria therapy, where successful treatments based on ancient recipes have been found, 261, there has already been some success in finding and testing ancient drugs and other treatments that are effective against AMR bacteria. 262. Computational community surveillance. One of the key tools identified by the WHO and others for the fight against rising antimicrobial resistance is improved surveillance of the spread and movement of AMR genes through different communities and regions. Recent advances in high-throughput DNA sequencing as a result of the Human Genome Project have resulted in the ability to determine the individual microbial genes in a sample. 263. Along with the availability of databases of known antimicrobial resistance genes, such as the Comprehensive Antimicrobial Resistance Database CARD 264, 265, and ResFinder 266, 267, this allows the identification of all the antimicrobial resistance genes within the sample, the so-called rhizostome. In doing so, a profile of these genes within a community or environment can be determined, providing insights into how antimicrobial resistance is spreading through a population and allowing for the identification of resistance that is of concern. 263. Phage therapy. Phage therapy is the therapeutic use of bacteriophages to treat pathogenic bacterial infections. 268. Phage therapy has many potential applications in human medicine as well as dentistry, veterinary science, and agriculture. 269. Phage therapy relies on the use of naturally occurring bacteriophages to infect and lyse bacteria at the site of infection in a host. Due to current advances in genetics and biotechnology, these bacteriophages can possibly be manufactured to treat specific infections. 270. Phages can be bioengineered to target multidrug-resistant bacterial infections, and their use involves the added benefit of preventing the elimination of beneficial bacteria in the human body. 31. Phages destroy bacterial cell walls and membrane through the use of lytic proteins which kill bacteria by making many holes from the inside out. 271. Bacteriophages can even possess the ability to digest the biofilm that many bacteria develop that protect them from antibiotics in order to effectively infect and kill bacteria. Bioengineering can play a role in creating successful bacteriophages. 271. Understanding the mutual interactions and evolutions of bacterial and phage populations in the environment of a human or animal body is essential for rational phage therapy. 272. Bacteriophagics are used against antibiotic-resistant bacteria in Georgia, George Eliava Institute, and in one institute in Wrocław, Poland. 273. 274. Bacteriophage cocktails are common drugs sold over the counter in pharmacies in Eastern countries. 275. 276. In Belgium, 4 patients with severe musculoskeletal infections received bacteriophage therapy with concomitant antibiotics. After a single course of phage therapy, no recurrence of infection occurred and no severe side effects related to the therapy were detected. 277. See also. Vector control. Vector control is any method to limit or eradicate the mammals, birds, insects or other arthropods, here collectively called vectors, which transmit disease pathogens. The most frequent type of vector control is mosquito control using a variety of strategies. Several of the neglected tropical diseases are spread by such vectors. Vector control taking place in the southern United States during the 1920s. Importance. For diseases where there is no effective cure, such as Zika virus, West Nile fever and dengue fever, vector control remains the only way to protect human populations. However, even for vector-borne diseases with effective treatments, the high cost of treatment remains a huge barrier to large amounts of developing world populations. Despite being treatable, malaria has by far the greatest impact on human health from vectors. In Africa, a child dies every minute of malaria. This is a reduction of more than 50% since 2000 due to vector control. 1. In countries where malaria is well established, the World Health Organization estimates countries lose 1.3% annual economic income due to the disease. 2. Both prevention through vector control and treatment are needed to protect populations. As the impacts of disease and virus are devastating, the need to control the vectors in which they carried is prioritized. Vector control in many developing countries can have tremendous impacts as it reduces mortality rates, especially among infants. 3. Because of the high movement of the population, disease spread is also a greater issue in these areas. 4. As many vector control methods are effective against multiple diseases, they can be integrated together to combat. As many vector control methods are effective against multiple diseases, they can be integrated together to combat multiple diseases at once. 5. The World Health Organization therefore recommends integrated vector management as the process for developing and implementing strategies for vector control. 6. Methods. Vector control focuses on utilizing preventive methods to control or eliminate vector populations. Common preventive measures are habitat and environmental control. Removing or reducing areas where vectors can easily breed can help limit their growth. For example, stagnant water removal, destruction of old tires and cans which serve as mosquito breeding environments, and good management of used water can reduce areas of excessive vector incidence. Further examples of environmental control is by reducing the prevalence of open defecation or improving the designs and maintenance of pit latrines. This can reduce the incidence of flies acting as vectors to spread diseases via their contact with feces of infected people. Reducing contact. Limiting exposure to insects or animals that are known disease vectors can reduce infection risks significantly. For example, bed nets, window screens on homes, or protective clothing can help reduce the likelihood of contact with vectors. To be effective, this requires education and promotion of methods among the population to raise the awareness of vector threats. Chemical control. Insecticides, larvicides, rodenticides, lethal ovitraps and repellents can be used to control vectors. For example, larvicides can be used in mosquito breeding zones, insecticides can be applied to house walls or bed nets, and use of personal repellents can reduce incidence of insect bites and thus infection. The use of pesticides for vector control is promoted by the World Health Organization (WHO) and has proven to be highly effective. 7. Biological control. The use of natural vector predators, such as bacterial toxins or botanical compounds, can help control vector populations. Using fish that eat mosquito larvae. The use of catfish to eat up mosquito larvae in ponds can eradicate the mosquito population, or reducing breeding rates by introducing sterilized male tsetse flies have been shown to control vector populations and reduce infection risks. 8. Legislation. United States. In the United States, cities or special districts are responsible for vector control. For example, in California, the Greater Los Angeles County Vector Control District is a special district set up by the state to oversee vector control in multiple cities. Plant breeding is the science of changing the traits of plants in order to produce desired characteristics. 1. It is used to improve the quality of plant products for use by humans and animals. 2. The goals of plant breeding are to produce crop varieties that boast unique and superior traits for a variety of applications. The most frequently addressed agricultural traits are those related to biotic and abiotic stress tolerance, grain or biomass yield, and use quality characteristics such as taste or the concentrations of specific biological molecules, proteins, sugars, lipids, vitamins, fibers, and ease of processing, harvesting, milling, baking, malting, blending, etc. 3. The Ecoro wheat right cultivar is sensitive to salinity. Plants resulting from a hybrid cross with cultivar W4910 left show greater tolerance to high salinity. Plant breeding can be performed using many different techniques, ranging from the selection of the most desirable plants for propagation, to methods that make use of knowledge of genetics and chromosomes, to more complex molecular techniques. Genes in a plant are what determine what type of qualitative or quantitative traits it will have. Plant breeders strive to create a specific outcome of plants and potentially new plant varieties, and in the course of doing so, narrow down the genetic diversity of that variety to a specific few biotypes. 4. It is practiced worldwide by individuals such as gardeners and farmers, and by professional plant breeders employed by organizations such as government institutions, universities, crop-specific industry associations or research centers. International development agencies believe that breeding new crops is important for ensuring food security by developing new varieties that are higher yielding, disease-resistant, drought-tolerant or regionally adapted to different environments and growing conditions. 5. A recent study shows that without plant breeding, Europe would have produced 20% fewer arable crops over the last 20 years, consuming an additional 21.6 million hectares, 53 million acres, of land and emitting 4 billion tons, 3.9 times 109 long tons, 4.4 times 109 short tons, of carbon. 6. 7. Wheat species created from Morocco are currently being crossed with plants to create new varieties for northern France. Soybeans, which were previously grown predominantly in the south of France, are now grown in southern Germany. 6. 8. History. Plant breeding started with sedentary agriculture and particularly the domestication of the first agricultural plants, a practice which is estimated to date back 9,000 to 11,000 years. 9. Initially early farmers simply selected food plants with particular desirable characteristics, and employed these as progenitors for subsequent generations, resulting in an accumulation of valuable traits over time. Grafting technology had been practiced in China before 2000 BCE. 10. By 500 BCE grafting was well established and practiced. 11. Gregor Mendel (1822–84) is considered the father of genetics. His experiments with plant hybridization led to his establishing laws of inheritance. Genetics stimulated research to improve crop production through plant breeding. Selective breeding played a crucial role in the Green Revolution of the 20th century. Modern plant breeding is applied genetics, but its scientific basis is broader, covering molecular biology, cytology, systematics, physiology, pathology, entomology, chemistry, and statistics biometrics. It has also developed its own technology. Classical plant breeding. Selective breeding enlarged desired traits of the wild cabbage plant Brassica oleracea over hundreds of years, resulting in dozens of today's agricultural crops. Cabbage, kale, broccoli, and cauliflower are all cultivars of this plant. One major technique of plant breeding is selection, the process of selectively propagating plants with desirable characteristics and eliminating or culling those with less desirable characteristics. 12. Another technique is the deliberate interbreeding crossing of closely or distantly related individuals to produce new crop varieties or lines with desirable properties. Plants are crossbred to introduce traits, genes from one variety or line into a new genetic background. For example, a mildew-resistant pea may be crossed with a high-yielding but susceptible pea, the goal of the cross being to introduce mildew resistance without losing the high-yield characteristics. Progeny from the cross would then be crossed with the high-yielding parent to ensure that the progeny were most like the high-yielding parent, backcrossing. The progeny from that cross would then be tested for yield selection, as described above, and mildew resistance and high-yielding resistant plants would be further developed. Plants may also be crossed with themselves to produce inbred varieties for breeding. Pollinators may be excluded through the use of pollination bags. Classical breeding relies largely on homologous recombination between chromosomes to generate genetic diversity. The classical plant breeder may also make use of a number of in vitro techniques such as protoplast fusion, embryo rescue or mutagenesis, see below, to generate diversity and produce hybrid plants that would not exist in nature. Traits that breeders have tried to incorporate into crop plants include 1. Improved quality, such as increased nutrition, improved flavor, or greater beauty. 2. Increased yield of the crop. 3. Increased tolerance of environmental pressures, salinity, extreme temperature, drought. 4. Resistance to viruses, fungi and bacteria. 5. Increased tolerance to insect pests. 6. Increased tolerance of herbicides. 7. Longer storage period for the harvested crop before World War II. Garden's Catalog from 1902. Successful commercial plant breeding concerns were founded from the late 19th century. Garden's Agricultural Plant Breeders in England was established in the 1890s by John Garden, who was one of the first to commercialize new varieties of agricultural crops created through cross-pollination. 13. The firm's first introduction was the Abundance oat, an oat variety. 14. 15. It is one of the first agricultural grain varieties bred from a controlled cross, introduced to commerce in 1892. 14. 15. In the early 20th century, plant breeders realized that Gregor Mendel's findings on the non-random nature of inheritance could be applied to seedling populations produced through deliberate pollinations to predict the frequencies of different types. Wheat hybrids were bred to increase the crop production of Italy during the so-called battle for grain, 1925-1940. Heterosis was explained by George Harrison Shull. It describes the tendency of the progeny of a specific cross to outperform both parents. The detection of the usefulness of heterosis for plant breeding has led to the development of inbred lines that reveal a heterotic yield advantage when they are crossed. Maize was the first species where heterosis was widely used to produce hybrids. Statistical methods were also developed to analyze gene action and distinguish heritable variation from variation caused by environment. In 1933, another important breeding technique, cytoplasmic male sterility, CMS, developed in maize, was described by Marcus Morton Rhodes. CMS is a maternally inherited trait that makes the plant produce sterile pollen. This enables the production of hybrids without the need for labor-intensive detasseling. These early breeding techniques resulted in large yield increase in the United States in the early 20th century. Similar yield increases were not produced elsewhere until after World War II. The Green Revolution increased crop production in the developing world in the 1960s. After World War II. In vitro culture of Vitis grapevine, Geisenheim Grape Breeding Institute. Following World War II, a number of techniques were developed that allowed plant breeders to hybridize distantly related species and artificially induce genetic diversity. When distantly related species are crossed, plant breeders make use of a number of plant tissue culture techniques to produce progeny from otherwise fruitless mating. Interspecific and intergeneric hybrids are produced from a cross of related species or genera that do not normally sexually reproduce with each other. These crosses are referred to as wide crosses. For example, the cereal triticale is a wheat and rye hybrid. The cells in the plants derived from the first generation created from the cross contained an uneven number of chromosomes and as a result was sterile. The cell division inhibitor colchicine was used to double the number of chromosomes in the cell and thus allow the production of a fertile line. Failure to produce a hybrid may be due to pre- or post-fertilization incompatibility. If fertilization is possible between two species or genera, the hybrid embryo may abort before maturation. If this does occur, the embryo resulting from an interspecific or intergeneric cross can sometimes be rescued and cultured to produce a whole plant. Such a method is referred to as embryo rescue. This technique has been used to produce new rice for Africa, an interspecific cross of Asian rice Oryza sativa and African rice O. glaberrima. Hybrids may also be produced by a technique called protoplast fusion. In this case, protoplasts are fused, usually in an electric field. Viable recombinants can be regenerated in culture. Chemical mutagens like ethylmethane sulfonate (EMS) and dimethyl sulfate (DMS) radiation and transposons are used for mutagenesis. Mutagenesis is the generation of mutants. The breeder hopes for desirable traits to be bred with other cultivars, a process known as mutation breeding. Classical plant breeders also generate genetic diversity within a species by exploiting a process called somoclonal variation, which occurs in plants produced from tissue culture, particularly plants derived from callus. Induced polyploidy, and the addition or removal of chromosomes using a technique called chromosome engineering may also be used. Agricultural research on potato plants. When a desirable trait has been bred into a species, a number of crosses to the favored parent are made to make the new plant as similar to the favored parent as possible. Returning to the example of the mildew-resistant pea being crossed with a high-yielding but susceptible pea, to make the mildew-resistant progeny of the cross most like the high-yielding parent, the progeny will be crossed back to that parent for several generations, see backcrossing. This process removes most of the genetic contribution of the mildew-resistant parent. Classical breeding is therefore a cyclical process. With classical breeding techniques, the breeder does not know exactly what genes have been introduced to the new cultivars. Some scientists therefore argue that plants produced by classical breeding methods should undergo the same safety testing regime as genetically modified plants. There have been instances where plants bred using classical techniques have been unsuitable for human consumption. For example, the poison solanine was unintentionally increased to unacceptable levels in certain varieties of potato through plant breeding. New potato varieties are often screened for solanine levels before reaching the marketplace. Even with the very latest in biotech-assisted conventional breeding, incorporation of a trait takes an average of 7 generations for clonally propagated crops, 9 for self-fertilizing, and 17 for cross-pollinating. 17. Modern plant breeding. Modern plant breeding may use techniques of molecular biology to select, or in the case of genetic modification, to insert, desirable traits into plants. Application of biotechnology or molecular biology is also known as molecular breeding. Modern facilities in molecular biology are now used in plant breeding. Marker-assisted selection. Sometimes many different genes can influence a desirable trait in plant breeding. The use of tools such as molecular markers or DNA fingerprinting can map thousands of genes. This allows plant breeders to screen large populations of plants for those that possess the trait of interest. The screening is based on the presence or absence of a certain gene as determined by laboratory procedures, rather than on the visual identification of the expressed trait in the plant. The purpose of marker-assisted selection, or plant genome analysis, is to identify the location and function phenotype of various genes within the genome. If all of the genes are identified, it leads to genome sequence. All plants have varying sizes and lengths of genomes with genes that code for different proteins, but many are also the same. If a gene's location and function is identified in one plant species, a very similar gene likely can also be found in a similar location in another related species' genome. 18. Doubled haploidy and reverse breeding. Homozygous plants with desirable traits can be produced from heterozygous starting plants, if a haploid cell with the alleles for those traits can be produced, and then used to make a doubled haploid. The doubled haploid will be homozygous for the desired traits. Furthermore, two different homozygous plants created in that way can be used to produce a generation of F1 hybrid plants which have the advantages of heterozygosity and a greater range of possible traits. Thus, an individual heterozygous plant chosen for its desirable characteristics can be converted into a heterozygous variety, F1 hybrid, without the necessity of vegetative reproduction but as the result of the cross of two homozygous, doubled haploid lines derived from the originally selected plant. 19. This shortcut has been dubbed reverse breeding. 20. Plant tissue culturing can produce haploid or double haploid plant lines in generations. This cuts down the genetic diversity taken from that plant species in order to select for desirable traits that will increase the fitness of the individuals. Using this method decreases the need for breeding multiple generations of plants to get a generation that is homogeneous for the desired traits, thereby saving much time over the natural version of the same process. There are many plant tissue culturing techniques that can be used to achieve haploid plants, but microspore culturing is currently the most promising for producing the largest numbers of them. 18. Genetic modification. Genetic modification of plants is achieved by adding a specific gene or genes to a plant, or by knocking down a gene with RNAi, to produce a desirable phenotype. The plants resulting from adding a gene are often referred to as transgenic plants. If for genetic modification genes of the species or of a crossable plant are used under control of their native promoter, then they are called cisgenic plants. Sometimes genetic modification can produce a plant with the desired trait or traits faster than classical breeding because the majority of the plant's genome is not altered. To genetically modify a plant, a genetic construct must be designed so that the gene to be added or removed will be expressed by the plant. To do this, a promoter to drive transcription and a termination sequence to stop transcription of the new gene, and the gene or genes of interest must be introduced to the plant. A marker for the selection of transformed plants is also included. In the laboratory, antibiotic resistance is a commonly used marker. Plants that have been successfully transformed will grow on media containing antibiotics. Plants that have not been transformed will die. In some instances, markers for selection are removed by backcrossing with the parent plant prior to commercial release. The construct can be inserted in the plant genome by genetic recombination using the bacteria Agrobacterium tumefaciens or rhizogenes, or by direct methods like the gene gun or microinjection. Using plant viruses to insert genetic constructs into plants is also a possibility, but the technique is limited by the host range of the virus. For example, cauliflower mosaic virus (CAMV) only infects cauliflower and related species. Another limitation of viral vectors is that the virus is not usually passed on to the progeny, so every plant has to be inoculated. The majority of commercially released transgenic plants are currently limited to plants that have introduced resistance to insect pests and herbicides. Insect resistance is achieved through incorporation of a gene from Bacillus thuringiensis (BT) that encodes a protein that is toxic to some insects. For example, the cotton bollworm, a common cotton pest, feeds on BT cotton. It will ingest the toxin and die. Herbicides usually work by binding to certain plant enzymes and inhibiting their action. 21. The enzymes that the herbicide inhibits are known as the herbicide's target site. Herbicide resistance can be engineered into crops by expressing a version of target site protein that is not inhibited by the herbicide. This is the method used to produce glyphosate-resistant, Roundup-ready, crop plants. Genetic modification can further increase yields by increasing stress tolerance to a given environment. Stresses such as temperature variation are signaled to the plant via a cascade of signaling molecules which will activate a transcription factor to regulate gene expression. Overexpression of particular genes involved in cold acclimation has been shown to produce more resistance to freezing, which is one common cause of yield loss. 22. Genetic modification of plants that can produce pharmaceuticals and industrial chemicals, sometimes called farming, is a rather radical new area of plant breeding. 23. The debate surrounding genetically modified food during the 1990s peaked in 1999 in terms of media coverage and risk perception. 24. And continues today. For example, Germany has thrown its weight behind a growing European mutiny over genetically modified crops by banning the planting of a widely grown pest-resistant corn variety. 25. The debate encompasses the ecological impact of genetically modified plants, the safety of genetically modified food and concepts used for safety evaluation like substantial equivalence. Such concerns are not new to plant breeding. Most countries have regulatory processes in place to help ensure that new crop varieties entering the marketplace are both safe and meet farmers' needs. Examples include variety registration, seed schemes, regulatory authorizations for GM plants, etc. Breeding in the microbiome. Industrial breeding of plants has unintentionally altered how agricultural cultivars associate with their microbiome. 26. In maize, for example, breeding has altered the nitrogen cycling taxa required to the rhizosphere, with more modern lines recruiting less nitrogen-fixing taxa and more nitrifiers and denitrifiers. 27. Microbiomes of breeding lines showed that hybrid plants share much of their bacterial community with their parents, such as cucurbita seeds and apple shoot endophytes. 28, 29, 30. In addition, the proportional contribution of the microbiome from parents to offspring corresponds to the amount of genetic material contributed by each parent during breeding and domestication. 30. Phenotyping and artificial intelligence. As of 2020, machine learning, and especially deep machine learning, has recently become more commonly used in phenotyping. Computer vision using ML has made great strides and is now being applied to leaf phenotyping and other phenotyping jobs typically performed by human eyes. Pound et al., 2017 and Singh et al., 2016 are especially salient examples of early successful application and demonstration of the general usability of the process across multiple target plant species. These methods will work even better with large, publicly available open datasets. 31. Speed breeding. Speed breeding is introduced by Watson et al., 2018. Classical, human-performed phenotyping during speed breeding is also possible, using a procedure developed by Richard et al., 2015. As of 2020, it is highly anticipated that SB and automated phenotyping will, combined, produce greatly improved outcomes. See section phenotyping and artificial intelligence above. 31. Genomic selection, GS. The NGS platform has substantially declined the time and cost required for sequencing and facilitated SNP discovery in model and non-model plants. This in turn has led to employing large-scale SNP markers in genomic selection approaches which aim at predicting genomic breeding values, GEBVs, of genotypes in a given population. This method can increase the selection accuracy and decrease the time of each breeding cycle. It has been used in different crops such as maize, wheat, etc. 32-33. Participatory plant breeding. Participatory plant breeding (PPB) is when farmers are involved in a crop improvement program with opportunities to make decisions and contribute to the research process at different stages. 34, 35, 36 Participatory approaches to crop improvement can also be applied when plant biotechnologies are being used for crop improvement. 37 Local agricultural systems and genetic diversity are strengthened by participatory programs, and outcomes are enhanced by farmers' knowledge of the quality required and evaluation of the target environment. 38 A 2019 review of participatory plant breeding indicated that it had not gained widespread acceptance despite its record of successfully developing varieties with improved diversity and nutritional quality, as well as greater likelihood of these improved varieties being adopted by farmers. This review also found participatory plant breeding to have a better cost-benefit ratio than non-participatory approaches, and suggested incorporating participatory plant breeding with evolutionary plant breeding. 39. Evolutionary plant breeding. Evolutionary plant breeding describes practices which use mass populations with diverse genotypes grown under competitive natural selection. Survival in common crop cultivation environments is the predominant method of selection, rather than direct selection by growers and breeders. Individual plants that are favored under prevailing growing conditions, such as environment and inputs, contribute more seed to the next generation than less adapted individuals. 40 Evolutionary plant breeding has been successfully used by the Nepal National Gene Bank to preserve Landrasa diversity within Jumli marshy rice while reducing its susceptibility to blast disease. These practices have also been used in Nepal with bean land races. 41. In 1929, Harlan and Martini proposed a method of plant breeding with heterogeneous populations by pooling an equal number of F2 seeds obtained from 378 crosses among 28 geographically diverse barley cultivars. In 1938, Harlan and Martini demonstrated evolution by natural selection in mixed dynamic populations as a few varieties that became dominant in some locations almost disappeared in others, poorly adapted varieties disappeared everywhere. 42. Evolutionary breeding populations have been used to establish self-regulating plant pathogen systems. Examples include barley, where breeders were able to improve resistance to Rhynchosporium caecalis scald over 45 generations. 43. An evolutionary breeding project grew F5 hybrid bulk soybean populations on soil infested by the soybean cyst nematode and was able to increase the proportion of resistant plants from 5% to 40%. The International Center for Agricultural Research in the Dry Areas (ICARDA) evolutionary plant breeding is combined with participatory plant breeding in order to allow farmers to choose which varieties suit their needs in their local environment. 43. An influential 1956 effort by Koita Sunnison to codify this approach coined the term evolutionary plant breeding and concluded that 15 generations of natural selection are desirable to produce results that are competitive with conventional breeding. 44. Evolutionary breeding allows working with much larger plant population sizes than conventional breeding. 42. It has also been used in tandem with conventional practices in order to develop both heterogeneous and homogeneous crop lines for low-input agricultural systems that have unpredictable stress conditions. 45. Evolutionary plant breeding has been delineated into 4 stages. 40. Stage 1. Genetic diversity is created, for example by manual crosses of inbreeding species or mixing of cultivars and outcrossing species. Stage 2. Multiplication of seeds. Stage 3. Seeds of each cross are then mixed to produce the first generation of the composite cross population, CCP. The entire offspring is sown to grow and set seed. As the number of plants in the population increases, a proportion of the harvested seed is saved for sowing. Stage 4. The seed can be used for continued evolutionary plant breeding or as a starting point for a conventional breeding effort. Issues and concerns. Breeding and food security. Issues facing plant breeding in the future include the lack of arable land, increasingly harsh cropping conditions and the need to maintain food security, which involves being able to provide the world population with sufficient nutrition. Crops need to be able to mature in multiple environments to allow worldwide access, which involves solving problems including drought tolerance. It has been suggested that global solutions are achievable through the process of plant breeding, with its ability to select specific genes allowing crops to perform at a level which yields the desired results. 46. One issue facing agriculture is the loss of land races and other local varieties which have diversity that may have useful genes for climate adaptation in the future. 43. Conventional breeding intentionally limits phenotype plasticity within genotypes and limits variability between genotypes. 45. Uniformity does not allow crops to adapt to climate change and other biotic stresses and abiotic stresses. 43. Plant breeders' rights. Plant breeders' rights is an important and controversial issue. Production of new varieties is dominated by commercial plant breeders, who seek to protect their work and collect royalties through national and international agreements based in intellectual property rights. The range of related issues is complex. In the simplest terms, critics of the increasingly restrictive regulations argue that, through a combination of technical and economic pressures, commercial breeders are reducing biodiversity and significantly constraining individuals, such as farmers, from developing and trading seed on a regional level. 47. Efforts to strengthen breeders' rights, for example, by lengthening periods of variety protection, or ongoing. Intellectual property legislation for plants often uses definitions that typically include genetic uniformity and unchanging appearance over generations. These legal definitions of stability contrast with traditional agronomic usage, which considers stability in terms of how consistent the yield or quality of a crop remains across locations and over time. 40. As of 2020, regulations in Nepal only allow uniform varieties to be registered or released. Evolutionary plant populations and many land races are polymorphic and do not meet these standards. 41. Environmental stressors. Uniform and genetically stable cultivars can be inadequate for dealing with environmental fluctuations and novel stress factors. 40. Plant breeders have focused on identifying crops which will ensure crops perform under these conditions. A way to achieve this is finding strains of the crop that has resistance to drought conditions with low nitrogen. It is evident from this that plant breeding is vital for future agriculture to survive, as it enables farmers to produce stress-resistant crops, hence improving food security. 48. In countries that experience harsh winters such as Iceland, Germany and further east in Europe, plant breeders are involved in breeding for tolerance to frost, continuous snow cover, frost drought desiccation from wind and solar radiation under frost, and high moisture levels in soil in winter. 49. Long-term process. Breeding is not a quick process, which is especially important when breeding to ameliorate a disease. The average time from human recognition of a new fungal disease threat to the release of a resistant crop for that pathogen is at least 12 years. 17. 50. Maintaining specific conditions. When new plant breeds or cultivars are bred, they must be maintained and propagated. Some plants are propagated by asexual means while others are propagated by seeds. Seed-propagated cultivars require specific control over seed source and production procedures to maintain the integrity of the plant breed's results. Isolation is necessary to prevent cross-contamination with related plants or the mixing of seeds after harvesting. Isolation is normally accomplished by planting distance, but in certain crops, plants are enclosed in greenhouses or cages, most commonly used when producing F1 hybrids. Nutritional value. Modern plant breeding, whether classical or through genetic engineering, comes with issues of concern, particularly with regard to food crops. The question of whether breeding can have a negative effect on nutritional value is central in this respect. Although relatively little direct research in this area has been done, there are scientific indications that, by favoring certain aspects of a plant's development, other aspects may be retarded. A study published in the Journal of the American College of Nutrition in 2004, entitled Changes in USDA Food Composition Data for 43 Garden Crops, 1950 to 1999, compared nutritional analysis of vegetables done in 1950 and in 1999, and found substantial decreases in 6 of 13 nutrients measured, including 6% of protein and 38% of riboflavin. Reductions in calcium, phosphorus, iron and ascorbic acid were also found. The study, conducted at the Biochemical Institute, University of Texas at Austin, concluded in summary, we suggest that any real declines are generally most easily explained by changes in cultivated varieties between 1950 and 1999, in which there may be trade-offs between yield and nutrient content. 51. Plant breeding can contribute to global food security as it is a cost-effective tool for increasing nutritional value of forage and crops. Improvements in nutritional value for forage crops from the use of analytical chemistry and rumen fermentation technology have been recorded since 1960. This science and technology gave breeders the ability to screen thousands of samples within a small amount of time, meaning breeders could identify a high-performing hybrid quicker. The genetic improvement was mainly in vitro dry matter digestibility (IVDMD), resulting in 0.7–2.5% increase. At just 1% increase in IVDMD, a single Bos taurus, also known as beef cattle, reported 3.2% increase in daily gains. This improvement indicates plant breeding is an essential tool in gearing future agriculture to perform at a more advanced level. 52. Yield. With an increasing population, the production of food needs to increase with it. It is estimated that a 70% increase in food production is needed by 2050 in order to meet the declaration of the World Summit on Food Security. But with the degradation of agricultural land, simply planting more crops is no longer a viable option. New varieties of plants can in some cases be developed through plant breeding that generate an increase of yield without relying on an increase in land area. An example of this can be seen in Asia, where food production per capita has increased twofold. This has been achieved through not only the use of fertilizers, but through the use of better crops that have been specifically designed for the area. 53. 54. Role of plant breeding in organic agriculture. Some critics of organic agriculture claim it is too low yielding to be a viable alternative to conventional agriculture in situations when that poor performance may be the result in part of growing poorly adapted varieties. 55. 56. It is estimated that over 95% of organic agriculture is based on conventionally adapted varieties, even though the production environments found in organic versus conventional farming systems are vastly different due to their distinctive management practices. 56. Most notably, organic farmers have fewer inputs available than conventional growers to control their production environments. Breeding varieties specifically adapted to the unique conditions of organic agriculture is critical for this sector to realize its full potential. This requires selection for traits such as 56. Water use efficiency. Nutrient use efficiency, particularly nitrogen and phosphorus, weed competitiveness. Tolerance of mechanical weed control. Pest disease resistance. Early maturity is a mechanism for avoidance of particular stresses, abiotic stress tolerance, i.e. drought, salinity, etc. Currently, few breeding programs are directed at organic agriculture, and until recently those that did address this sector have generally relied on indirect selection, i.e. selection in conventional environments for traits considered important for organic agriculture. However, because the difference between organic and conventional environments is large, a given genotype may perform very differently in each environment due to an interaction between genes and the environment. See gene-environment interaction. If this interaction is severe enough, an important trait required for the organic environment may not be revealed in the conventional environment, which can result in the selection of poorly adapted individuals. 55. To ensure the most adapted varieties are identified, advocates of organic breeding now promote the use of direct selection, i.e. selection in the target environment, for many agronomic traits. There are many classical and modern breeding techniques that can be utilized for crop improvement in organic agriculture despite the ban on genetically modified organisms. For instance, controlled crosses between individuals allow desirable genetic variation to be recombined and transferred to seed progeny via natural processes. Marker-assisted selection can also be employed as a diagnostics tool to facilitate selection of progeny who possess the desired traits, greatly speeding up the breeding process. 57. This technique has proven particularly useful for the introgression of resistance genes into new backgrounds, as well as the efficient selection of many resistance genes pyramided into a single individual. Molecular markers are not currently available for many important traits, especially complex ones controlled by many genes. List of Animal breeding is a branch of animal science that addresses the evaluation, using best linear unbiased prediction and other methods, of the genetic value, estimated breeding value, EBV, of livestock. Selecting for breeding animals with superior EBV in growth rate, egg, meat, milk, or wool production, or with other desirable traits has revolutionized livestock production throughout the entire world. The scientific theory of animal breeding incorporates population genetics, quantitative genetics, statistics, and recently molecular genetics, and is based on the pioneering work of Sewell Wright, J. Lush, and Charles Henderson. Breeding stock. Breeding stock is a group of animals used for the purpose of planned breeding. When individuals are looking to breed animals, they look for certain valuable traits in purebred animals, or may intend to use some type of crossbreeding to produce a new type of stock with different, and presumably superior abilities in a given area of endeavor. For example, when breeding swine for meat, the breeding stock should be sound, fast-growing, muscular, lean, and reproductively efficient. 1. The subjective selection of breeding stock in horses has led to many horse breeds with particular performance traits. 2. While breeding animals is common in an agricultural setting, it is also a common practice for the purpose of selling animals meant as pets, such as cats, dogs, horses, and birds, as well as less common animals, such as reptiles or some primates. Purebred breeding. Mating animals of the same breed for maintaining such breed is referred to as purebred breeding. Opposite to the practice of mating animals of different breeds, purebred breeding aims to establish and maintain stable traits that animals will pass to the next generation. By breeding the best to the best, employing a certain degree of inbreeding, considerable culling, and selection for superior qualities, one could develop a bloodline or breed superior in certain respects to the original base stock. Such animals can be recorded with a breed registry, the organization that maintains pedigrees and/or stud books. The observable phenomenon of hybrid vigor stands in contrast to the notion of breed purity. For laboratory purposes, organisms such as mice have been inbred to 100% pure lines, as offered for sale by the Jackson Laboratory. But this is highly unusual and difficult to do for most organisms. 2003, archived from the Animal breeding. Animal breeding is a branch of animal science that addresses the evaluation, using best linear unbiased prediction and other methods, of the genetic value, estimated breeding value, EBV, of livestock. Selecting for breeding animals with superior EBV in growth rate, egg, meat, milk, or wool production, or with other desirable traits has revolutionized livestock production throughout the entire world. The scientific theory of animal breeding incorporates population genetics, quantitative genetics, statistics, and recently molecular genetics, and is based on the pioneering work of Sewell Wright, J. Lush, and Charles Henderson. Breeding stock. Breeding stock is a group of animals used for the purpose of planned breeding. When individuals are looking to breed animals, they look for certain valuable traits in purebred animals, or may intend to use some type of crossbreeding to produce a new type of stock with different, and presumably superior abilities in a given area of endeavor. For example, when breeding swine for meat, the breeding stock should be sound, fast-growing, muscular, lean, and reproductively efficient. 1. The subjective selection of breeding stock in horses has led to many horse breeds with particular performance traits. 2. While breeding animals is common in an agricultural setting, it is also a common practice for the purpose of selling animals meant as pets, such as cats, dogs, horses, and birds, as well as less common animals, such as reptiles or some primates. Purebred breeding. Mating animals of the same breed for maintaining such breed is referred to as purebred breeding. Opposite to the practice of mating animals of different breeds, purebred breeding aims to establish and maintain stable traits that animals will pass to the next generation. By breeding the best to the best, employing a certain degree of inbreeding, considerable culling, and selection for superior qualities, one could develop a bloodline or breed superior in certain respects to the original base stock. Such animals can be recorded with a breed registry, the organization that maintains pedigrees and/or stud books. The observable phenomenon of hybrid vigor stands in contrast to the notion of breed purity. For laboratory purposes, organisms such as mice have been inbred to 100% pure lines, as offered for sale by the Jackson Laboratory. But this is highly unusual and difficult to do for most organisms. 2003, archived from the original, on the 21st of October 2008, implementing the Global Plan of Action for Animal Genetic Resources, /info/ Animal husbandry. Animal husbandry is the branch of agriculture concerned with animals that are raised for meat, fiber, milk, or other products. It includes day-to-day care, management, production, nutrition, selective breeding, and the raising of livestock. Husbandry has a long history, starting with the Neolithic Revolution when animals were first domesticated, from around 13,000 BC onwards, predating farming of the first crops. During the period of ancient societies like ancient Egypt, cattle, sheep, goats, and pigs were being raised on farms. Cattle feedlot in Colorado, United States. Major changes took place in the Columbian Exchange, when Old World livestock were brought to the New World, and then in the British agricultural revolution of the 18th century, when livestock breeds like the Dishley Longhorn cattle and Lincoln Longwool sheep were rapidly improved by agriculturalists, such as Robert Bakewell, to yield more meat, milk, and wool. A wide range of other species, such as horse, water buffalo, llama, rabbit, and guinea pig, are used as livestock in some parts of the world. Insect farming, as well as aquaculture of fish, mollusks, and crustaceans, is widespread. Modern animal husbandry relies on production systems adapted to the type of land available. Subsistence farming is being superseded by intensive animal farming in the more developed parts of the world, where, for example, beef cattle are kept in high-density feedlots, and thousands of chickens may be raised in broiler houses or batteries. On poorer soil, such as in uplands, animals are often kept more extensively and may be allowed to roam widely, foraging for themselves. Animal agriculture at modern scale drives climate change, ocean acidification, and biodiversity loss. Most livestock are herbivores, except, among the most commonly kept species, for pigs and chickens which are omnivores. Ruminants like cattle and sheep are adapted to feed on grass. They can forage outdoors or may be fed entirely or in part on rations richer in energy and protein, such as pelleted cereals. Pigs and poultry cannot digest the cellulose in forage and require other high-protein foods. Etymology. The verb to husband, meaning, to manage carefully, derives from an older meaning of husband, which in the 14th century referred to the ownership and care of a household or farm, but today means the control or judicious use of resources, and in agriculture, the cultivation of plants or animals. Farmers and ranchers who raise livestock are considered to practice animal husbandry. History. Birth of husbandry. The domestication of ruminants, like these fat-tailed sheep in Afghanistan, provided nomads across the Middle East and Central Asia with a reliable source of food. The domestication of livestock was driven by the need to have food on hand when hunting was unproductive. The desirable characteristics of a domestic animal are that it should be useful to the domesticator, should be able to thrive in his or her company, should breed freely, and be easy to tend. Domestication was not a single event, but a process repeated at various periods in different places. Sheep and goats were the animals that accompanied the nomads in the Middle East, while cattle and pigs were associated with more settled communities. The first wild animal to be domesticated was the dog. Half-wild dogs, perhaps starting with young individuals, may have been tolerated as scavengers and killers of vermin, and being naturally pack hunters, were predisposed to become part of the human pack and join in the hunt. Prey animals, sheep, goats, pigs and cattle, were progressively domesticated early in the history of agriculture. Pigs were domesticated in the Near East between 8,500 and 8,000 BC, 4 sheep and goats in or near the Fertile Crescent about 8,500 BC, 5 and cattle from wild aurochs in the areas of modern Turkey and Pakistan around 8,500 BC. 6. A cow was a great advantage to a villager as she produced more milk than her calf needed, and her strength could be put to use as a working animal, pulling a plow to increase production of crops, and drawing a sledge, and later a cart, to bring the produce home from the field. Draft animals were first used about 4000 BC in the Middle East, increasing agricultural production immeasurably. 3. In southern Asia, the elephant was domesticated by 6000 BC. 7. Fossilized chicken bones dated to 5040 BC have been found in northeastern China, far from where their wild ancestors lived in the jungles of tropical Asia, but archaeologists believe that the original purpose of domestication was for the sport of cockfighting. 8. Meanwhile, in South America, the llama and the alpaca had been domesticated, probably before 3000 BC, as beasts of burden and for their wool. Neither was strong enough to pull a plow, which limited the development of agriculture in the New World. 3. Horses occur naturally on the steppes of Central Asia, and their domestication began around 3000 BC in the Black Sea and Caspian Sea region. Although horses were originally seen as a source of meat, their use as pack animals and for riding followed. Around the same time, the wild ass was being tamed in Egypt. Camels were domesticated soon after this, 9th with the Bactrian camel in Mongolia and the Arabian camel becoming beasts of burden. By 1000 BC, caravans of Arabian camels were linking India with Mesopotamia and the Mediterranean. 3. Ancient civilizations. Milking cattle in ancient Egypt. In ancient Egypt, cattle were the most important livestock, and sheep, goats, and pigs were also kept. Poultry including ducks, geese, and pigeons were captured in nets and bred on farms, where they were force-fed with dough to fatten them. 10th, the Nile provided a plentiful source of fish. Honeybees were domesticated from at least the Old Kingdom, providing both honey and wax. 11. In ancient Rome, all the livestock known in ancient Egypt were available. In addition, rabbits were domesticated for food by the 1st century BC. To help flush them out from their burrows, the polecat was domesticated as the ferret, its use described by Pliny the Elder. 12. Medieval husbandry. Shepherd with sheep in woven hurdle pen. Medieval France. 15th century, Bodleian Library, MS Deuce 195. In Northern Europe, agriculture including animal husbandry went into decline when the Roman Empire collapsed. Some aspects such as the herding of animals continued throughout the period. By the 11th century, the economy had recovered and the countryside was again productive. 13. The Domesday Book recorded every parcel of land and every animal in England. There was not one single hide, nor a yard of land, nay, moreover, not even an ox, nor a cow, nor a swine was there left, that was not set down in the King's Writ. 14. For example, the royal manor of Earley in Berkshire, one of thousands of villages recorded in the book, had in 1,086 inches 2 fisheries worth paying tax of 7s and 60 each year, and 20 acres of meadow for livestock. Woodland for feeding, 70 pigs. 15. The improvements of animal husbandry in the medieval period in Europe went hand in hand with other developments. Improvements to the plow allowed the soil to be tilled to a greater depth. Horses took over from oxen as the main providers of traction. New ideas on crop rotation were developed and the growing of crops for winter fodder gained ground. 16. Peas, beans and vetches became common. They increased soil fertility through nitrogen fixation, allowing more livestock to be kept. 17. Columbian exchange. Exploration and colonization of North and South America resulted in the introduction into Europe of such crops as maize, potatoes, sweet potatoes and manioc, while the principal Old World livestock, cattle, horses, sheep and goats, were introduced into the New World for the first time along with wheat, barley, rice and turnips. 18. Agricultural Revolution. The Lincoln Longwool breed was improved by Robert Bakewell in the 18th century. Selective breeding for desired traits was established as a scientific practice by Robert Bakewell during the British Agricultural Revolution in the 18th century. One of his most important breeding programs was with sheep. Using native stock, he was able to quickly select for large, yet fine-boned sheep, with long, lustrous wool. The Lincoln Longwool was improved by Bakewell and in turn the Lincoln was used to develop a subsequent breed, named the New or Dishley Leicester. It was hornless and had a square, meaty body with straight top lines. 19. These sheep were exported widely and have contributed to numerous modern breeds. Under his influence, English farmers began to breed cattle for use primarily as beef. Longhorned heifers were crossed with the Westmoreland bull to create the Dishley Longhorn. 20. The semi-natural, unfertilized pastures formed by traditional agricultural methods in Europe were managed by grazing and mowing. As the ecological impact of this land management strategy is similar to the impact of such natural disturbances as grazing and wildfire, this agricultural system shares many beneficial characteristics with a natural habitat, including the promotion of biodiversity. This strategy is declining in Europe today due to the intensification of agriculture. The mechanized and chemical methods used are causing biodiversity to decline. 21. Practices. Systems. Herdwick sheep in an extensive hill farming system, Lake District, England. Traditionally, animal husbandry was part of the subsistence farmer's way of life, producing not only the food needed by the family but also the fuel, fertilizer, clothing, transport and draft power. Killing the animal for food was a secondary consideration, and wherever possible its products such as wool, eggs, milk and blood by the Maasai were harvested while the animal was still alive. 22. In the traditional system of transhumance, people and livestock moved seasonally between fixed summer and winter pastures. In montane regions the summer pasture was up in the mountains, the winter pasture in the valleys. 23. Animals can be kept extensively or intensively. Extensive systems involve animals roaming at will, or under the supervision of a herdsman, often for their protection from predators. Ranching in the western United States involves large herds of cattle grazing widely over public and private lands. 24. Similar cattle stations are found in South America, Australia and other places with large areas of land and low rainfall. Ranching systems have been used for sheep, deer, ostrich, emu, llama and alpaca. 25. In the uplands of the United Kingdom, sheep are turned out on the fells in spring and graze the abundant mountain grasses untended, being brought to lower altitudes late in the year, with supplementary feeding being provided in winter. 26. In rural locations, pigs and poultry can obtain much of their nutrition from scavenging, and in African communities, hens may live for months without being fed, and still produce 1 or 2 eggs a week. 22. Pigs in an intensive system, Midwestern United States. At the other extreme, in the more developed parts of the world, animals are often intensively managed. Dairy cows may be kept in zero grazing conditions with all their forage brought to them. Beef cattle may be kept in high-density feedlots. 27. Pigs may be housed in climate-controlled buildings and never go outdoors. 28. Poultry may be reared in barns and kept in cages as laying birds under lighting-controlled conditions. In between these two extremes are semi-intensive, often family-run farms where livestock graze outside for much of the year. Silage or hay is made to cover the times of year when the grass stops growing, and fertilizer, feed, and other inputs are brought onto the farm from outside. 29. Feeding. Cattle around an outdoor feeder. Animals used as livestock are predominantly herbivorous, the main exceptions being the pig and the chicken which are omnivorous. The herbivores can be divided into concentrate selectors, which selectively feed on seeds, fruits and highly nutritious young foliage, grazers, which mainly feed on grass, and intermediate feeders, which choose their diet from the whole range of available plant material. Cattle, sheep, goats, deer and antelopes are ruminants. They digest food in 2 steps, chewing and swallowing in the normal way, and then regurgitating the semi-digested cud to chew it again and thus extract the maximum possible food value. 30. The dietary needs of these animals is mostly met by eating grass. Grasses grow from the base of the leaf blade, enabling it to thrive even when heavily grazed or cut. 31. In many climates grass growth is seasonal, for example in the temperate summer or tropical rainy season, so some areas of the crop are set aside to be cut and preserved, either as hay dried grass or as silage fermented grass. 32. Other forage crops are also grown and many of these, as well as crop residues, can be ensiled to fill the gap in the nutritional needs of livestock in the lean season. 33. Cattle feed pellets of pressed linseed. Extensively reared animals may subsist entirely on forage, but more intensively kept livestock will require energy and protein-rich foods in addition. Energy is mainly derived from cereals and cereal byproducts, fats and oils and sugar-rich foods, while protein may come from fish or meat meal, milk products, legumes and other plant foods, often the byproducts of vegetable oil extraction. 34. Pigs and poultry are non-ruminants and unable to digest the cellulose in grass and other forages, so they are fed entirely on cereals and other high-energy foodstuffs. The ingredients for the animals' rations can be grown on the farm or can be bought, in the form of pelleted or cubed, compound foodstuffs specially formulated for the different classes of livestock, their growth stages and their specific nutritional requirements. Vitamins and minerals are added to balance the diet. 35. Farmed fish are usually fed pelleted food. 35. Breeding. The breeding of farm animals seldom occurs spontaneously but is managed by farmers with a view to encouraging traits seen as desirable. These include hardiness, fertility, docility, mothering abilities, fast growth rates, low feed consumption per unit of growth, better body proportions, higher yields, and better fiber qualities. Undesirable traits such as health defects and aggressiveness are selected against. 36-37. Selective breeding has been responsible for large increases in productivity. For example, in 2007, a typical broiler chicken at 8 weeks old was 4.8 times as heavy as a bird of similar age in 1957, 36. While in the 30 years to 2007, the average milk yield of a dairy cow in the United States nearly doubled. 36. Animal health. Vaccinating a goat, Niger. Good husbandry, proper feeding, and hygiene are the main contributors to animal health on the farm, bringing economic benefits through maximized production. When, despite these precautions, animals still become sick, they are treated with veterinary medicines, by the farmer and the veterinarian. In the European Union, when farmers treat their own animals, they are required to follow the guidelines for treatment and to record the treatments given. 38. Animals are susceptible to a number of diseases and conditions that may affect their health. Some, like classical swine fever 39 and scrapie 40, are specific to one type of stock, while others, like foot and mouth disease affect all cloven-hoofed animals. 41. Animals living under intensive conditions are prone to internal and external parasites. Increasing numbers of sea lice are affecting farmed salmon in Scotland. 42. Reducing the parasite burdens of livestock results in increased productivity and profitability. 43. Where the condition is serious, governments impose regulations on import and export, on the movement of stock, quarantine restrictions and the reporting of suspected cases. Vaccines are available against certain diseases, and antibiotics are widely used where appropriate. At one time, antibiotics were routinely added to certain compound foodstuffs to promote growth, but this practice is now frowned on in many countries because of the risk that it may lead to antimicrobial resistance in livestock and in humans. 44. Familiar livestock, Incan. Watercolor drawing of a farmyard with cow, horse, pigs, and chickens, 1869. Governments are concerned with zoonoses, diseases that humans may acquire from animals. Wild animal populations may harbor diseases that can affect domestic animals which may acquire them as a result of insufficient biosecurity. An outbreak of Nipah virus in Malaysia in 1999 was traced back to pigs becoming ill after contact with fruit-eating flying foxes, their feces and urine. The pigs in turn passed the infection to humans. 45. Avian flu H5N1 is present in wild bird populations and can be carried large distances by migrating birds. This virus is easily transmissible to domestic poultry, and to humans living in close proximity with them. Other infectious diseases affecting wild animals, farm animals and humans include rabies, leptospirosis, brucellosis, tuberculosis and trichinosis. 46. Range of species. There is no single universally agreed definition of which species are livestock. Widely agreed types of livestock include cattle for beef and dairy, sheep, goats, pigs, and poultry. Various other species are sometimes considered livestock, such as horses, 47, while poultry birds are sometimes excluded. In some parts of the world, livestock includes species such as buffalo, and the South American camelids, the alpaca and llama. 48, 49, 50. Some authorities use much broader definitions to include fish in aquaculture, micro-livestock such as rabbits and rodents like guinea pigs, as well as insects from honeybees to crickets raised for human consumption. 51. Shearing a merino sheep for its wool. Products. Animals are raised for a wide variety of products, principally meat, wool, milk, and eggs, but also including tallow, isinglass and rennet. 52, 53. Animals are also kept for more specialized purposes, such as to produce vaccines, 54, and antiserum containing antibodies for medical use. 55. Where fodder or other crops are grown alongside animals, manure can serve as a fertilizer, returning minerals and organic matter to the soil in a semi-closed organic system. 56. Branches. Dairy. A modern rotary milking parlor, Germany. Although all mammals produce milk to nourish their young, the cow is predominantly used throughout the world to produce milk and milk products for human consumption. Other animals used to a lesser extent for this purpose include sheep, goats, camels, buffaloes, yaks, reindeer, horses and donkeys. 57. All these animals have been domesticated over the centuries, being bred for such desirable characteristics as fecundity, productivity, docility and the ability to thrive under the prevailing conditions. Whereas in the past cattle had multiple functions, modern dairy cow breeding has resulted in specialized Holstein Friesian type animals that produce large quantities of milk economically. Artificial insemination is widely available to allow farmers to select for the particular traits that suit their circumstances. 58. Whereas in the past cows were kept in small herds on family farms, grazing pastures and being fed hay in winter, nowadays there is a trend towards larger herds, more intensive systems, the feeding of silage and zero grazing, a system where grass is cut and brought to the cow, which is housed year-round. 59. In many communities, milk production is only part of the purpose of keeping an animal, which may also be used as a beast of burden or to draw a plow, or for the production of fiber, meat and leather, with the dung being used for fuel or for the improvement of soil fertility. Sheep and goats may be favored for dairy production in climates and conditions that do not suit dairy cows. 57. Meat. The Hereford is a hardy breed of beef cattle, now raised in many countries around the world. Meat, mainly from farmed animals, is a major source of dietary protein and essential nutrients around the world, averaging about 8% of man's energy intake. 60. The actual types eaten depend on local preferences, availability, cost and other factors, with cattle, sheep, pigs and goats being the main species involved. Cattle generally produce a single offspring annually, which takes more than a year to mature. Sheep and goats often have twins and these are ready for slaughter in less than a year. Pigs are more prolific, producing more than 1 litter of up to about 61 piglets each year. 62. Horses, donkeys, deer, buffalo, llamas, alpacas, guanacos and vicuñas are farmed for meat in various regions. Some desirable traits of animals raised for meat include fecundity, hardiness, fast growth rate, ease of management and high food conversion efficiency. About half of the world's meat is produced from animals grazing on open ranges or on enclosed pastures, the other half being produced intensively in various factory farming systems. These are mostly cows, pigs or poultry, and often reared indoors, typically at high densities. 63. Poultry. Battery hens, Brazil. Poultry, kept for their eggs and for their meat, include chickens, turkeys, geese and ducks. The great majority of laying birds used for egg production are chickens. Methods for keeping layers range from free-range systems, where the birds can roam as they will but are housed at night for their own protection, through semi-intensive systems where they are housed in barns and have perches, litter and some freedom of movement, to intensive systems where they are kept in cages. The battery cages are arranged in long rows in multiple tiers, with external feeders, drinkers, and egg collection facilities. This is the most labor-saving and economical method of egg production but has been criticized on animal welfare grounds as the birds are unable to exhibit their normal behaviors. 64. In the developed world, the majority of the poultry reared for meat is raised indoors in big sheds, with automated equipment under environmentally controlled conditions. Chickens raised in this way are known as broilers, and genetic improvements have meant that they can be grown to slaughter weight within 6 or 7 weeks of hatching. Newly hatched chicks are restricted to a small area and given supplementary heating. Litter on the floor absorbs the droppings and the area occupied is expanded as they grow. Feed and water is supplied automatically and the lighting is controlled. The birds may be harvested on several occasions or the whole shed may be cleared at one time. 65. A similar rearing system is usually used for turkeys, which are less hardy than chickens, but they take longer to grow and are often moved on to separate fattening units to finish. 66. Ducks are particularly popular in Asia and Australia and can be killed at 7 weeks under commercial conditions. 67. Aquaculture. Freshwater fish farming, France. Aquaculture has been defined as the farming of aquatic organisms including fish, mollusks, crustaceans and aquatic plants and implies some form of intervention in the rearing process to enhance production, such as regular stocking, feeding, protection from predators, etc. Farming also implies individual or corporate ownership of the stock being cultivated. 68. In practice it can take place in the sea or in freshwater, and be extensive or intensive. Whole bays, lakes or ponds may be devoted to aquaculture, or the farmed animal may be retained in cages, fish, artificial reefs, racks or strings, shellfish. Fish and prawns can be cultivated in rice paddies, either arriving naturally or being introduced, and both crops can be harvested together. 69. Fish hatcheries provide larval and juvenile fish, crustaceans and shellfish, for use in aquaculture systems. When large enough these are transferred to growing on tanks and sold to fish farms to reach harvest size. Some species that are commonly raised in hatcheries include shrimps, prawns, salmon, tilapia, oysters and scallops. Similar facilities can be used to raise species with conservation needs to be released into the wild, or game fish for restocking waterways. Important aspects of husbandry at these early stages include selection of breeding stock, control of water quality and nutrition. In the wild, there is a massive amount of mortality at the nursery stage. Farmers seek to minimize this while at the same time maximizing growth rates. 70. Insects. Crickets being raised for human consumption, Thailand. Bees have been kept in hives since at least the first dynasty of Egypt, 5,000 years ago, 71, and man had been harvesting honey from the wild long before that. Fixed comb hives are used in many parts of the world and are made from any locally available material. 72. In more advanced economies, where modern strains of domestic bee have been selected for docility and productiveness, various designs of hive are used which enable the combs to be removed for processing and extraction of honey. Quite apart from the honey and wax they produce, honeybees are important pollinators of crops and wild plants, and in many places hives are transported around the countryside to assist in pollination. 73. Sericulture, the rearing of silkworms, was first adopted by the Chinese during the Shang dynasty. 74. The only species farmed commercially is the domesticated silkmoth. When it spins its cocoon, each larva produces an exceedingly long, slender thread of silk. The larvae feed on mulberry leaves and in Europe, only one generation is normally raised each year as this is a deciduous tree. In China, Korea and Japan however, 2 generations are normal, and in the tropics, multiple generations are expected. Most production of silk occurs in the Far East, with a synthetic diet being used to rear the silkworms in Japan. 75. Insects form part of the human diet in many cultures. 76. In Thailand, crickets are farmed for this purpose in the north of the country, and palm weevil larvae in the south.